Hi everyone. We'll just wait for a few minutes and see who all else joins, and then we'll get started. Okay, I think we can get started. I'll just share my screen. You guys let me know if you can see it. Hmm. Can everyone see my screen?
Yes.
Okay, great. So I think we can go ahead now. Right. So, hi everyone. Today we're going to be discussing some problems and topics related to the coursework that was uploaded for week 3. This mainly involves DNA replication. I'm covering more of the prokaryotic DNA replication in this session. I have some questions relating to eukaryotic as well later on, but I thought I'd cover these topics in more detail, at least now. Um, yeah, and last week we covered mitosis and the different phases of mitosis. And one of the phases in mitosis or before mitosis is interphase, and that is um, a very lengthy period of the cell cycle, the longest one. And part of that is S phase. And S phase, as we discussed last time, is where duplication of DNA or replication of DNA takes place. And for that, you need G1 as a preparatory phase. Um, and So now, obviously, if DNA is being replicated, we need to know how it's being done, right? So that's how we're sort of linking this to the next part, which is in S phase, how is the DNA being replicated? Okay. Uh, so you have all already seen the process of replication, and you know that there is a leading strand and a lagging strand when it comes to the replication fork. So you know that on the lagging strand, there is these formation of these fragments, one after another, called the Okazaki fragments. And these are formed because DNA replication, the addition of these DNTPs, is going to only take place from the 5' to 3' direction. That means onto the 3' end of a primer, you will add the DNA polymerase enzyme will be able to add on a DNTP only on the 3' end, meaning that the growth of the strand is going to take place from 5' to 3' direction. But DNA, the helix is anti-parallel, right? So how does that happen? If both strands are, in opposite directions, how can DNA replication take place only from 5' to 3'? So that's why um, there is a requirement of one of the strands to have these shorter fragments called Okazaki fragments that are later then sort of ligated together to finally form uh, the copy of the lagging strand. And this was discovered uh, by two professors, a husband-wife couple, both of whose surname is Okazaki. And they did a very elegant experiment called which is known as a pulse chase experiment. And you guys have definitely heard of pulse chase experiments before, I think, when it comes to, the figuring out how different proteins are transported through the cell, or how different organelles process certain proteins. A lot of studies with respect to the Golgi apparatus and ER have been done using pulse chase labeling. These days, like before, it used to be done with like radioactive isotopes. Now people have even figured out ways to do it with fluorescence labeling, and then they can actually, with microscopy, look at localization of different um, say proteins in a cell, maybe while it's developing. I've seen works done in flatworms where they've looked at how different proteins that are involved in its development and growth and regeneration have been fluorescently labeled initially as a pulse, and then a different protein that's either unlabeled or labeled with a different color is then flooded or given in excess to that cell or to that organism, and then that is chased. So essentially what is happening in a pulse chase experiment is you give a period of pulsing where you expose the cell or organism, whatever you're working with some pulse labeled compound. So in this case, in the Okazaki experiment, they have this E. coli that was pulse labeled with this H3 thymidine, okay, at a lower temperature. And after that, you do what is called a chase. That means essentially you allow it to recover. In that time, you allow it to go about its natural course. And then you can isolate DNA, you can isolate proteins from the cells during this chase, after the chase time is done, and then look at how essentially the labeled moiety has distributed itself. So here with pulse labeling, they did it for initially a 20-second pulse, and then, or maybe it was 10 seconds. And then after that, yeah, they followed it with a 2-minute chase, and then they had a longer chase as well. So this one, A, this is where you have pulse labeling, and then you have chasing, and then you have a longer chasing. So what did they see, right? Because before this, it was not known that Okazaki fragments exist. They thought people were trying to figure out how it is that the other the other lagging strand is synthesized at all. And so they saw that after labeling, they saw that when they did density gradient centrifugation, they saw that in the fraction where they are taking out this 1000 to 2000 nucleotide, like where you can get 1000 to 2000 length nucleotide sequences, they see that there are these radio labeled DNA fragments. So now if you are duplicating an entire length of the gene of E. coli, why do you have these short nucleotide stretches in the first place? What are these doing, and how come they've incorporated this radio-labeled dye while the DNA is replicating, right? So is the DNA fragmenting? And that's actually something that these professors, some criticism that they'd faced initially when they had published their work and presented it at a conference. Other professors and other people in the field were and understandably saying that, okay, maybe this is just some artifact in your work. Maybe some DNA degraded, and that's why you have shorter nucleotide sequences. What's to say that this is something new? So then they actually had to validate this extensively in a whole bunch of other different types of assays and experiments to prove that no, this is not degrading. Actually, these are genuine 1000 to 2000 um, nucleotide long DNA fragments. And then eventually these were published and known to be these Okazaki fragments. So yeah, that's basically what it is. And so once they prove that these Okazaki fragments exist, and eventually, because you'll see that after chasing the pulse after for quite a long time, you see that this spike corresponds to basically the radio labeled um, thymidine spiking in the DNA. And then over time, they see that this pulse is going down. This is for the fragments that are short. These are specifically for the short fragments, which were found in this fractions 5 to 7. And I'll explain a little more about density gradient centrifugation so you understand what these fractions mean. Okay. So essentially, there are different fractions in which you can get different size DNA fragments based on how you centrifuge them, right? So in this fraction, we are seeing that initially there is a spike, makes sense, we have pulsed it. Then we chase it for some time, this pulse starts going down. And then after a point, it completely goes away, meaning that these were integrated together somehow. So that's how they came up that these DNA fragments actually were not only being synthesized separately, but after synthesis was done, these fragments were actually incorporating into a long and continuous chain of genomic DNA. So they posited their discontinuous replication mechanism for the lagging strand. So that's the Okazaki experiment. And oh, I didn't go into detail about. So I'll tell you about density gradient centrifugation in a little bit. I think that's in a later slide. So I just put some questions up here, very simple ones to start us off. And then I'll talk a little bit about the Messerson-Stahl experiment, which was one of, the very sort of benchmark experiments that set up a lot of understanding of how DNA replicates and what mode it replicates in. So here on the right, you can see that you have a strand of DNA essentially, and it's made of the nucleotide subunits consisting of your 5-carbon sugar here, and then you have your phosphate group on C5, and you have your nucleotide subunit on C1, and these are joined. One subunit is joined to the next subunit with the help of this sugar phosphate backbone that's created here. So the phosphodiester linkage over here is what attaches one to the next. So this is the 3' or the C3 carbon, and this is the C5 carbon, and this is where essentially if you are adding one DNTP to a growing chain, this is where this reaction takes place, and I'll talk about it in more detail later on. And of course, you can have either pyrimidines or purines attached to your C1. So the first question, and as you guys know, I will read the question and you guys can call out the answer. So what is the directionality of the two strands in a DNA double helix? Easy one. Do you guys know?
Option C.
Option C.
That's right. So that's great. So they run anti-parallel. You can see here that one is running 5 dash to 3 dash, right? So because why is it 5 dash to 3 dash? Because 5 dash may, on this C5, on this end, you have a free phosphate. And here on the 3 dash, you have a free OH. And that's what allows this growth to take place. So that's why it's running from 5 dash to 3 dash. And naturally, the DNA strand over here would run in the opposite direction. So anti-parallel to it. And this is a very easy question. So the Watson-Crick base pairing rules, which of these base pairs is correct?
Option B.
Yeah,
B.
great. Yeah, that's absolutely right. A pairs with T and G pairs with C. And as you know, A and T have two bonds, two hydrogen bonds, and G and C have three hydrogen bonds. And the number of hydrogen bonds can actually between two bases can actually influence how much effort or how much energy it takes to break that bond. And this is important for later, right? Okay, so now we're coming to this famous Michelson and Stahl experiment. So this was explained in great detail in your coursework as well, but it's so important, I thought I'd go over it again to remind you guys so that we can nicely tackle the questions after this, right? So what these professors, Michelson and Stahl in 1958, they performed this experiment to essentially rule out any other posited forms, any other posited modes of DNA replication that people had, come up with. That, oh, okay, it's conservative or it's dispersive, but they wanted to show whether it's semi-conservative or not. So how did they do this, right? They they took some initially the DNA within E. coli. It was treated with this N15. It's a heavy nitrogen, and it was labeled with that. So that means all of this E. coli now has this N labeled, 15N labeled DNA, right? And N is a major component of the purines and pyrimidines, right? So it's going to incorporate into the DNA as the cell takes it up and incorporates it while it's dividing and producing its DNA. So eventually, after say culturing E. coli in 15N media for long enough, you'll have all of its DNA having incorporated this 15N. Okay. And then what they did is these cells, then they added it to 14N, so regular nitrogen supplemented media. And then they grew it for a couple of generations. And then at each generation, they performed this cesium chloride ultracentrifugation, density gradient ultracentrifugation, essentially meaning that you have a density gradient formed by this solution of cesium chloride, where you have different sizes of DNA based on the gradient and density of cesium chloride at each point in this tube. You will have different sizes of DNA, and in this case it's different densities of DNA, sorry, which means how much of this heavy nitrogen it has incorporated. They will be separated out accordingly. So you will have your N14 higher up and you'll have your N15 higher up. So the more you have this heavy isotope, the more it will go down in density, and the more you have the lighter one, the higher up it will be. And so in this elegant way, as you know, E. coli divides every 20 minutes. So every 20 minutes, you have a new generation. So every 20 minutes, they took cells, they lysed, they took the DNA out, they centrifuged it in this density gradient method, and then they ran it to see, okay, what is going on? Why are there so many different bands? Okay. So at generation 0, I think I have another slide for this. Yeah, so at generation 0, where you have only got this 15N labeled E. coli, so the DNA from that will be fully heavy, right? Both strands fully heavy. Then they looked at the first generation and they see that there is an intermediate heavy, like a hybrid of these light and heavy. So it's sort of intermediate. And then they looked at the second generation and they see that there's a mix. You have both the hybrid between heavy and light, and also you have completely light. And then they move to generation 3, and generation 4. And they see that the hybrid is diluting out slowly and you have much more of this light labeled DNA. So that indicates that, there's definitely a period of time where you have a hybrid forming, indicating that you have this initial molecule being split in half, the two strands are being taken apart, and you have a fresh strand being synthesized on the existing strand. And then that would obviously be the same for the next generation and the next. So eventually you will have completely N14 labeled DNA and just a little bit, which is hardly detectable, of this hybrid. And so how did this rule out the other ones, conservative and dispersive, right? So for conservative, what was the, yeah, so this is, I've written it in a little more detail. So in conservative, as you can see here, the theory was that the parental strands actually remain together. So actually, they don't separate out and they don't act as templates for one another. And so you form a completely new daughter DNA helix somehow, presumably by magic. um, and then that keeps going. So you always have one parent DNA helix that will give rise to a completely separate one without separating any strands. So there's no mixing of parent and daughter strands. Okay, but how can we rule that out over here? They saw in the first generation that there was a hybrid. So that immediately rules out this point altogether. There should be here either HH, heavy, heavy, or light, light, but not an intermediate, right? So conservative is out now. That's not happening. What about the other one? So there is dispersive. So each in this theory, people had said that, okay, maybe the parental strand breaks into some smaller strands. And these are used to make fresh strands that are then all joined together later. So it's all mixed up, right? So how can we rule this one out from the Michelson-Stahl experiment, right? Oops. So here, for the dispersive one, then what we would have seen is that this hybrid, because there essentially we're saying that there's a constant mixing of the heavy and light, or the parent and the daughter strands. So there would have been a constant hybrid HL or heavy light that would have been seen for many generations. And you would not see this majority of light light all just by the time the second generation comes, because here also there would have been mixing. So here, because they got a clear light light band, that means that there is no such huge amount of mixing that the dispersive model suggests. And so that's how the Michelson and Stahl experiment ruled out the conservative and dispersive mode of DNA replication and went ahead with what was initially posited but not well proved, that DNA replication is semi-conservative, right? So does anyone have any questions till now about this experiment or the Okazaki experiment? Hopefully everyone has understood the concepts. But if there's anything else, you can let me know. You can stop me in between if you're confused. Yeah. Right. So now I had said that I would tell you about density gradient centrifugation because it is a very, very useful technique even now. And it's not just for DNA. You can use it for cells. I use it for cells all the time. You can use it for proteins. It just depends on what kind of density gradient media you use and what gradient you require for separating out different fractions of proteins or DNA or even cells based on their size or density. Okay. So here, this is an image from a very old paper, very famous paper actually, this one, where they have separated out nuclear DNA or nuke DNA and mitochondrial DNA. And this is one of the big tubes where you have this density gradient media that's put in. It's an ultracentrifuge tube, and you place it inside the rotor and you spin it really fast. So under very high centrifugal force, or in an ultracentrifuge, it really goes above. I think ultracentrifugation starts from beyond 20,000 to 25,000 RCF or Gs. So all the way up to like 100,000, based on what you're separating, you can use that high. A solution of cesium chloride molecules under high centrifugal forces, they actually dissociate. Then what happens is these heavy cesium plus atoms, they'll be forced away from the center and towards the outer end of the tube. But this at the same time diffuses back towards the top of the tube, and this forms a density gradient. And the DNA molecules that are placed in this gradient, you will essentially layer it on from the top here. They will migrate to the point where that DNA molecule or that strand of DNA has the same density as the gradient in that layer. So essentially, you have layers of different density cesium chloride, and your DNA is sort of moving through it from the top like this. And suppose over here it's found the same density, then it will remain here. It won't go beyond it. And this gradient is enough to separate out different types of DNA. You can separate out based on their GC content. So it's quite a linear relation between GC content and density. So that's easy to figure out or calculate using density gradient ultracentrifugation. Or you can have physical form of your DNA. So like linear versus circular molecules, you can separate them out um, based on that as well. And in this experiment, what they've done is they centrifuged at, look how high it is, 450,000 G, right? So what does this into G mean? You guys know, right? That this is like the relative centrifugal force. So essentially 1 G is, right, how much gravity is. So this is that many times that. So it's the force felt by the sample, and that's how high the force is. Um, so they did that for 10 whole hours, and they saw that there are these two distinct bands from this shared linear nuclear DNA, and below that they have the circular mitochondrial DNA. So these are two different physical forms of DNA that they have separated. And obviously, because they had allowed some ethidium bromide to intercalate with the DNA at the time, they were able to visualize it using UV light. Yeah. So that's how essentially you can separate them out, and then you can, there are tubes that have like a hole that you can poke here and then start separating out the fractions. You know how you have size exclusion chromatography? It's a bit reminiscent to that you have essentially, you're collecting fractions one by one and you're running them to see how the different fractions look, and you can have these calibration curves that you can create. And people do this for cells to separate out dead cells from live cells, debris from healthy cells. People do it for proteins. People do it for viruses. When we purify viruses, you can actually ultracentrifuge them and use a density gradient to um, separate out viruses from the media. When you're producing, say, lentiviruses or retroviruses, you can use that as well. So it's a very, very useful lab technique. And it was used a lot back then, a lot more, I think, back then, for all of these different preparations of different forms of DNA and so on and so forth. Yeah. So that is that. Now we have a few more questions. Then we can talk about these people on the right side. So this question is, which of the following was not a proposed model for DNA replication? Uh, do answer if you know. I hope you all can hear me.
Option D.
That's correct. Someone has messaged C, so that's not right, right? Because semi-conservative is the one that it actually is. Because I just explained the Michelson-Stahl experiment where they showed quite irrefutably that DNA replication is semi-conservative. So the parent strand acts as a template for the next generation strand or the daughter strand. So yeah, transformative model, that's like not a thing. So it's the other three: conservative, dispersive, and semi-conservative, after which the real one turned out to be semi-conservative. Yeah, great. Right. So in the conservative model of DNA replication, what happens to the parental strands? So I explained this. Please read the options and let me know what you guys think. Anyone, any idea? So take it from the word conservative. What would that imply, right? That something is being conserved. It's not being changed, it's not being distributed, right?
Option B.
That sounds good. Does anyone have any other answer? Anything to contest this answer? No, we all agree. Yeah, so that's completely right. It is option B. So the parental strands were posited that, okay, they stay together and a completely new DNA molecule is formed. So that was, of course, disproved. And finally, which scientist first proposed the semi-conservative model?
Option A.
Hmm.
Option B.
So the answer is option A, actually. It was Watson and Crick who initially posited this when they didn't have experimental proof, like they didn't have completely irrefutable experimental proof like this experiment. But because they had sort of figured out how the DNA structure is, and they had seen these repeating units, they had thought, okay, there seems to be complementarity, so there's a possibility that there is a semi-conservative nature in DNA replication. And they had posited that, they had hypothesized that. But Michelson and Stahl were the ones to actually perform the experiments. But the people who proposed it were Watson and Crick. So in relation to that, these guys here, this is Michelson and Stahl. This is them. Uh, More recent, obviously, colored image. And up here, this is Watson and Crick. So I thought you could just put a face to the name while we study and read about them. Yeah. So hopefully this cleared up any questions about this important experiment. I'll move on to a few more questions, right, about it. What was the key element used by Michelson and Stahl to differentiate the parental DNA from the newly synthesized DNA?
Option B.
Option B.
That's right. Great. So it was the heavy nitrogen isotope N15, which was which they allowed the E. coli to initially they maintain the E. coli in essentially at the start to allow it to incorporate into the DNA. Okay, what was the method? Okay, this is now you know very well. Used to separate DNA based on its density in the Michelson-Stahl experiments.
Option B.
Yeah, great. Yeah, so that one I yeah explained for you guys. Now, after one round of replication in N14 media, what kind of DNA was observed by Michelson and Stahl?
Option D.
That's correct. Does anyone have any other answer that they thought it might be? Then that's correct. Yeah. So if you have one DNA, like one whatever, one strand of DNA, then the next in the very first round of replication after the G0 stage, you're going to have a completely hybrid stage where you have both of the parent strands entwined with fresh light strands or daughter strands. Yeah. And finally, the results of this experiment ruled out which models of DNA replication. Again, this you'll be able to answer very easily.
Option C.
Mm, Maybe try again. Which did it rule out? So it basically proved that it's not those.
Option B.
Yeah, you got it right. So conservative and dispersive, these were models that people had hypothesized at the time, but because they had this hybrid form that they saw in the first generation in the experiment, they could rule out that conservative and dispersive were models of DNA replication that were valid. Yeah, so the answer is B. Great. Okay, so you guys learned about DNA replication and all the different factors involved, right? The DNA poly, holoenzyme, all of its different elements, and visualizing it is quite hard sometimes. So I found this nice video. I thought we could all watch it and then go to some more questions after that. So I'll just play it and please let me know if you can hear it.
DNA replication is the process by which a duplicate copy of the
Can everyone hear the video?
genetic material is made.
Can everyone hear the video?
Yes,
Yes,
ma'am.
ma'am.
Okay, okay, great. All right, so let's just watch it and then we can discuss after that.
DNA replication is the process by which a duplicate copy of the genetic material is made before a cell divides. The steps in replication are highly conserved in all organisms. There are a few details that differ between bacteria and eukaryotes, but we focus on bacterial replication in this animation for simplicity. The process of DNA replication is broadly described as having three phases: initiation, elongation, and termination. Let's begin by considering the initiation phase. For copying to take place, the double helical structure of the DNA must first be opened at a site termed the origin of replication to allow access by the machinery that will copy the DNA. This opening is regulated by a series of initiation regulatory proteins not shown here. Once open, a DNA helicase, a ring of 6 subunits, is loaded onto one strand of the DNA at each side of the replication bubble. This establishes two replication forks that will move away from each other as replication proceeds. The synthesis of DNA is catalyzed by an enzyme called DNA polymerase. However, DNA polymerases cannot start a new strand from scratch. Instead, they can only elongate an existing polymer of DNA or RNA. Consequently, a primer sequence needs to be made from which DNA polymerase can continue its synthesis. Primers are made by an enzyme called primase. Once access to the single strands of the DNA has been obtained, the primase is loaded onto the DNA. The primase synthesizes a small stretch of RNA by copying the sequence of nucleotides on the template strand. A DNA polymerase will later elongate this small RNA stretch. The next steps allow loading of the DNA polymerases onto the DNA to copy the entire chromosome. The first component that is loaded is the sliding clamp, a ring-shaped structure that binds at the 3' end of the newly made RNA primer. To get onto DNA and encircle it, the clamp is loaded by a clamp loader complex. The 5-protein clamp loader complex binds to the sliding clamp, and binding of ATP allows opening of the sliding clamp ring. The opened ring then encircles the template primer junction at the 3' end of the primer. Interaction with the 3' end of the DNA stimulates ATP hydrolysis, and the clamp loader dissociates from the DNA to be replaced by the replicative DNA polymerase. The 3' end of the primer is positioned in the polymerase active site to allow addition of nucleotides to this end. During elongation, the replication bubble gets bigger as the helicases move apart, establishing two replication forks that move in opposite directions. The sliding clamp and polymerase unit at each replication fork moves along the DNA as the helicase unwinds the double-stranded DNA to expose single strands. Let's focus on just the right-hand fork for the moment, as the processes at both forks are identical. Polymerases are loaded onto both the top and the bottom DNA strands at the fork. Because polymerases only synthesize DNA in the 5' to 3' direction, the two strands must be copied in opposite directions. The bottom strand in this fork is the leading strand. The polymerase on this strand moves continuously from left to right, synthesizing DNA as it travels. The top strand is the lagging strand, on which just a short stretch of DNA is made by the polymerase elongating in the 5' to 3' direction. After synthesizing this short stretch of DNA, the polymerase then dissociates and a new polymerase binds at the fork to elongate the next RNA primer. So priming and elongation are happening repeatedly on the lagging strand as the replication fork moves along the DNA. Synthesis is discontinuous. The short stretches of DNA that are made on the lagging strand are called Okazaki fragments after their discoverer. During the course of replication, these fragments are stitched together. When the lagging strand polymerase runs up against a previously made Okazaki fragment while in the process of synthesizing DNA, the replicative DNA polymerase III is replaced by DNA polymerase I, which has 5' to 3' exonuclease activity. This exonuclease then degrades the RNA primer in front of it, synthesizing DNA as it goes. Once the RNA primer is removed, the two sections of DNA are joined by DNA ligase. Now the lagging strand has been elongated by one more Okazaki fragment. While this view of the replication fork is easiest to understand, in reality the leading and lagging polymerases are coupled and move in concert with the helicase. This coupling requires one DNA strand to be looped around so that the lagging polymerase can synthesize DNA in the 5' to 3' direction while moving with the fork. We explore this coupling in more detail in a separate animation. Much less is known about the details of the termination of DNA replication. Once the entire chromosome is copied, the two replication forks meet each other and are dismantled. The ends of the new DNA strands are then joined, again by DNA ligase. The two daughter DNA molecules now consist of one parent strand of DNA shown in grey and one newly made strand shown in green. Thus, DNA replication is termed semi-conservative.
Great. So I hope that was educational for you guys and that this recapped what you've learned from the coursework from week 3. If you have any questions, please, you can stop me at any time. Yeah, so this is just sort of visualizing what you saw. So as you know, in E. coli or prokaryotes, you're going to have a double-stranded and circular DNA or a covalently closed circular DNA, that's called. And when you have these replisomes that are replicating in bidirectionally, you have this loop form, and then they sort of both reach the opposite end and terminate there, and then it's ligated together in the new strand. And this is how the complex looks with your leading strand template over here and the new strand coming in red. And similarly here you have the lagging strand with your new Okazaki fragments. And meanwhile you have another thing happening, which is your primase, which is important for making your primers. So it makes these RNA primers based on the template DNA, so short RNA primers that allow DNA polymerase to then attach and add DNTPs to the growing strand. And this is your helicase, important for unwinding DNA. And your clamp loader and sliding clamp as well, that are important for loading. So the clamp loader, I think it loads the sliding clamp onto the primer template hybrid duplex, right? And the sliding clamp itself allows the DNA polymerase unit to be to move along the DNA double-stranded helix without it, and in the single-stranded regions without it falling off. So it helps with the improving the processivity, or how long it can keep synthesizing fresh strand without falling off. So these are the different elements to this DNA polymerase holoenzyme. And oh yeah, you have these guys, which are these single-stranded DNA binding proteins, or SSBPs. They are interesting. They are oligomerized. So you have many of them that sort of bind together to form an oligomer, and then they bind two single-stranded regions within the replication bubble so that the single-stranded regions don't bind back to each other or reanneal. So all of these different units come together for a very fast mechanism, almost 1,000 base pairs per second you can add on. In E. coli, it can add on in a new strand using all of these different units that work very, very fast together. And another interesting point that I had read about once was that, you have a lot of both in the case of, DNTPs as well as being added to a new strand, as well as, in fact, more in the case of amino acids being added on during translation to a growing polypeptide chain. How come it's so fast, right? Because at any given time, here it could be an A, or it could be a T, or it could be a G, or it could be a C. And in the case of amino acids, you have like 20 of them. Oops. So any one of them could bind. So statistically saying, you have here a 1 in 4 chance that one could bind, and in amino acids, a 1 in 20. So how is it so fast when you have all these different molecules that have to basically try out and then bind, and then, okay, if it's the right one, then cool. If not, then find the one that's correct for this particular position, right? Because DNA polymerase is not going to bind the wrong nucleotide onto the, onto, say, it's not going to bind a T to a G, right? It's going to put the right one. So how is it so fast when all of this trial and error is required? So it turns out, actually, if you do the calculations, I haven't, but people have, that it sort of works out. The math works out, and despite there having to be some trial and error, it still can manage. So within those microseconds or milliseconds of time, all of this trial and error can take place. And you have to remember that it's not just one molecule or two molecules that are hanging out. It's many, many molecules that are constantly diffusing around. So you have these DNTPs, and they're all diffusing around, or these amino acids, they're all diffusing around, and they're all moving and sort of vibrating really fast. And so they're able to interact with this site. And regardless of the fact that there are so many choices for any one position when you are expanding a DNA sequence or a protein, it seems that it doesn't matter, and you can actually synthesize it in time. So I thought that's something interesting that you guys could think about, that how these things work out with respect to time, because otherwise we'd spend forever replicating our DNA, or the E. coli would take like 3 hours, but it just takes 20 minutes. So that's really crazy. Right. So with that, let's move on to some more questions. So here is one, which is, what is the main enzyme that's responsible for addition of DNTPs?
Option B.
Yeah, that is correct. It is DNA polymerase. And bonus question, can it add DNTPs, or can it start a fresh fragment, or does it need something to polymerize or add DNTPs onto? Can DNA polymerase start a new strand on its own?
No, ma'am.
Yeah. So it would need a primer, right? So yeah, that is just something that is important to remember. Yeah. All right. So now DNA synthesis, we discussed this earlier, right? Which direction does it occur in? What do you guys think?
Option B.
Yeah, anyone else? Any other answers? That's correct then. Very good. 5 dash to 3 dash is correct. So is this 5 dash to 3 dash of the parent strand, or 5 dash to 3 dash of the daughter strand? Bonus question. What do you guys think? Think about it, what it looks like in your mind when you're thinking about DNA synthesis and how nucleotides are added on to a growing strand. Is it 5 dash to 3 dash of the new strand that's being formed, or 5 dash to 3 dash of the existing strand? Which direction are we really talking about?
Of the parent strand.
So that's not correct actually. So it's 5 dash to 3 dash uh, of the daughter strand. So your primer is binding and it has a 5' and then a 3', right? So it's going to elongate in the 3' end because that's where you add on the nucleotides. So it's of the okay, of the strand that's being created next to the primer, like continuing from the primer. So that was just the bonus question. All right, next one. In prokaryotic replication, what binds to the replication origin to initiate DNA synthesis?
Option C, DNA.
That's correct. That's very good. So DNA A protein, that's important for initiating, or the initiator, and it binds to an ET-rich region in the origin or URI in prokaryotic replication. So great. Okay. Here is a question that relates to the previous one. So what characteristic of the origin of replication helps it unwind easily?
Option C.
That's correct. So it's AT rich, meaning that it can be melted much easily, much more easily. Great. So the next question. So in prokaryotes, which replication in prokaryotes proceeds in which manner? Is it unidirectional, bidirectional, random directions from multiple origins?
Option B.
Option B.
Yeah, so you're right. Very good. So as you saw in earlier in the video and in the cartoons as well, it is moving bidirectionally until it meets at the other end. Yeah. And from multiple origins actually happens in eukaryotes, right? Because you have multiple origins at which you have these initiating factors that bind and allow replication to start so that you have such a long linear chromosome, it can actually be replicated within a relatively short period of time. So it's important to have multiple origins in eukaryotes. So that is not the case in prokaryotes. Yeah. But then again, because in prokaryotes it's shorter, it can replicate quick. Right. So the structure formed when DNA strands separate and the replication process starts. What is this called? This is just a simple one.
Option B.
Yeah. Great. So replication bubble. This is where you have some ssDNA that is created. And you have polymerase binding for either both the leading strand and the lagging strand. Right. Which enzyme is responsible for proofreading and error correction during DNA replication? What do you guys think?
Option B.
That's correct. That's very good. So that's what that's what DNA polymerase 3 does. It has proofreading and this exonuclease activity that helps for error correction during DNA replication. Okay, continuing from there, the next question is, what is the origin of replication in E. coli called?
Option D.
Option D.
Great, so you guys know this very well. It's option D. It's called OREC, and there is just the one per chromosome of a prokaryote. Okay, this is just a sort of facts question, but you guys know approximately how many nucleotide pairs an E. coli genome contains?
Option C.
That's correct. Yes, and also the person who is messaging, good job. It is C. 4.6 into 10 power 6, or 4.6 million. That's quite a lot that they have to duplicate in 20 minutes. Okay, and what speed do these replication forks in the E. coli progress forwards? Do you guys remember from your coursework and material?
Option A.
Anyone, any other answers? Anyone, any other ideas? No? Okay, so the answer is actually 500 to 1,000 nucleotides per second. So they can go up to 1,000 nucleotides per second. So that's because of the high processivity of DNA polymerase enzymes and all of its components that allow it to move very fast and accurately with the replication fork on the strand of DNA. And it makes sure, so high processivity means that essentially how long it can keep adding nucleotides to the growing strand without falling off the strand. So DNA Pol has high processivity and high accuracy. So, and oh yeah, of course, this should happen. This addition of nucleotides should happen without making mistakes or falling off. So it can go up to 1,000 nucleotides per second. So just imagine how fast all of these processes are taking place, that it can duplicate so much in that short period of time. So what protein is responsible for initiating DNA replication in E. coli? So
Option A.
that's right. It's DNA A. Very good. All right. So this I just thought I'd put here so that you guys can sort of visualize it a little while you're thinking about this complex and how it helps polymerize DNA. So do you guys remember the tau protein? So do you guys know what the function of the tau protein is? Anyone? So tau protein is,
Option
yeah, go ahead, go ahead.
Option D.
That's right. Very good. So it is option D, which is both A and C. So for those who may have forgotten, so the tau protein is an important component of this replisome mechanism machinery, right? And it does two things. One is that it connects DNA pol 3 to the clamp, and the clamp is very important for placing the DNA pol 3 in the correct place onto the single-stranded DNA and allowing it to move forward. So the clamp helps with improving processivity and accuracy of DNA pol 3's activity. And it also helps stimulate helicase activity. So when it is bound and helicase is in contact, just to make sure that helicase is not just, acting anywhere, only where this tau protein is, when it binds to helicase, it can stimulate its activity. So only in specific regions where required, helicase will then cause unwinding of the DNA and create a single-stranded DNA contained in a replication bubble. So that's what the tau protein does, and it's a very important component of the DNA Pol holoenzyme complex. Right. So this is a easy question. Helicase is also called which of these?
Option B.
That would be correct. Does anyone have any conflicting answers? Everyone's correct. In the comments also correct. Great. So yeah, DNA is your initiator proteins, which we have covered. DNA B is helicase, and we'll come to the other ones. So I won't spoil it now. All right, so which enzyme unwinds the DNA double helix? Easy one. What do you guys think? Yeah, in the comments, okay, in the comments, you may have confused this a little bit. Unwinding the double helix, or essentially creating single-stranded regions, that is the
Option
job. Yeah, go ahead.
Option D.
That is correct. Yes, very good. So, yeah, I was explaining that, yeah, topoisomerases, they do help with maintaining single-stranded regions, or they maintain like not like untangling of the DNA. So they help with making sure that these sort of supercoiled condensed regions of DNA don't interfere with the replication process. So they help with making sure that there's not too much torsion stress in the DNA. So they do that, but they don't unwind the DNA. They don't have that catalytic activity that helicase does for this process. So yeah, this answer was option D, helicase, DNA helicase. Right. So now this one. This is a little bit complicated to read, but DNA primase is a what? So what do you guys think is the correct answer?
Option C.
Hmm. Does anyone have any other answer?
Option B.
Option B. So C and B have both been positive. Does anyone have any other answer? Okay, Samiksha is saying D. So does anyone say A? No. Okay, so the actual answer is D. Okay, so the person in the comments, you're right. It is a DNA dependent RNA polymerase. So I'll explain why that is the case. So DNA primase is important for what? Right? It's important for making the primers that are then used for, then the DNA polymerase use that as a sort of starting point to add DNTPs to. So it is made of RNA. So the first thing is that if you're making a primer out of RNA, then that is an RNA polymerase, correct? If you are synthesizing, so DNA primase can synthesize a primer from scratch. It does not need from scratch in the sense it needs a template, but it does not need any kind of primer of its own. So on a template strand in a specified region, the DNA primase can sit and create a short RNA primer. And then why am I saying it's option D? Why is it DNA dependent? Because it needs the DNA template. So the DNA template has the sequence that it needs to form the complementary primer to, such that then it forms this RNA primer onto which then the DNTPs can be added on. So DNA primase therefore becomes a DNA dependent RNA polymerase. So I hope that is now clear to everybody. Right? If anyone has any confusions. Okay, great. So, okay. So I was saying holoenzyme a whole bunch of times, right? So I thought I would just, explain what a holoenzyme is to anyone who may have forgotten, right? So we were saying that DNA polymerase is a holoenzyme. It's a holoenzyme complex consisting of many different things. But a holoenzyme does not necessarily mean that you have many different proteins or subunits, right? Any enzyme can have that. What it means here is the difference between an apoenzyme and a holoenzyme. So apoenzyme is a protein component of an enzyme which is inactive until it binds to some sort of cofactor, and that cofactor is typically not a protein. And holoenzyme is this complete active enzyme that consists of an apoenzyme, which is the protein part, which was inactive, and then once it's bound, its necessary cofactor. So this whole holoenzyme is now active and catalytically active. It's ready to do whatever function it is supposed to. And here in this case, the example is DNA polymerase, where without magnesium, which is the cofactor, you have apoenzymes. You have the apoenzyme DNA pol, and then the holoenzyme is when you have magnesium that binds to, I think, the alpha subunit of DNA polymerase. And this actually allows it, makes it catalytically active and allows it to perform DNA synthesis. So that is the holoenzyme in this case. And there are many different um, enzymes during, I think then during respiration, though I can't exactly remember. And maybe I can tell you guys next time. And during translation, RNA polymerization also, you have all of these different enzymes that require different cofactors. You guys must have studied about PCR as well, right? And in PCR, the importance of magnesium comes through. So what is PCR, right? It's a polymerase chain reaction where you have, you provide a template DNA, you provide some primers, you put in a polymerase, which is a Taq polymerase typically, which is basically an enzyme that will polymerize and form these new strands on top of continuing from where your primers bind. But one very important component that we always make sure to add or already have incorporated in some buffer or something is magnesium ions. So without that, your polymerase is not going to work. So we always include magnesium ions in our buffers. And the opposite is true if you want to make sure that your polymerases or any other enzyme that requires divalent cations for their functioning, you don't want them to function. What can you do to stop the reaction? You can add this chelating agent called EDTA, right? So you guys must have heard of EDTA. So EDTA is chelates divalent cations like magnesium and calcium. It forms these sort of coordination interactions with them. And we can add EDTA to maybe a reaction or into different enzyme into different say you don't want like it even nucleases and stuff, RNases, DNases, they require these ions as well. So if you want to make sure that none of them are able to function, you will make sure that EDTA is there. That prevents the functioning of these various different cleaving enzymes. So that's a practical sort of application of these cofactors and how we add them artificially when we're doing PCR or other experiments. Yeah. And a little more into detail, I found this, I think someone answered a question and I like the way they explained it. So this is the sort of binding site or active pocket of DNA polymerase. So DNA polymerase has one main active catalytic site. And here you can see these D or aspartic acid residues that are helpful for binding with these magnesium ions, these metal ions or divalent cation type of ions. And you will have this entire addition of DNTP to the existing strand is catalyzed over here. So let me break it down. So you have your dsDNA here, and then this is your template strand, and you're going to add in a whole new DNTP here. So how is that happening? We need to create a phosphodiester linkage over here. So you have an OH in the previous one and you have this phosphate here. And so what needs to happen essentially is a nucleophilic attack. But you already have, as this person has mentioned, you have this huge amount of electrons from this triphosphate. So it's like a huge cloud of electrons that is essentially almost sterically hindering the oxygen from being able to reach there and attack the phosphate. Right? So what do we do? How do we solve that problem? How does it get there and nucleophilically attack this phosphate, right? This alpha phosphate over here of the triphosphate on the sugar. So we have these magnesiums that are bound at this active site onto these aspartic acid residues. So you know that aspartic acid is a negatively charged amino acid. It's an acidic one. So you have an O minus that it will help with coordinating with this metal ion, and that essentially starts drawing the electrons towards itself. Not completely, it just sort of pulls the density towards it and polarizes that such that the oxygen, it's not exactly how it's happening, but it's a nice way to visualize it. So it pulls the electron cloud a little bit towards itself and allows the oxygen to sort of access this phosphorus and make the nucleophilic attack that allows it to form the phosphodiester linkage. So I thought that's a nice way to sort of envision what's going on inside this catalytic pocket of DNA polymerase and how the cofactor magnesium is important for this process, right? So I hope that is clear for everyone. Yeah, so with that, I think we can move to last couple of questions. 1 second. 24. Oh, okay. Right. So I have a couple more questions here. So what is the role of DNA-C in replication? So we have covered DNA-A, B, G as primates, but what is DNA-C? You guys remember?
Option A.
Someone is saying option E. Does anyone have any other answers? Everyone is happy with this. So you're right. It is indeed DNAC. It's important. It's a helicase loader, and it's important for loading helicase onto the single-stranded DNA. Great. What is the shape of the helicase protein? So you'd remember the video that your professor had shown. It was really nice. You guys remember?
Option C.
That is right. Very good. So it's a hexameric ring. So it has these 6 different subunits that are arranged in this ring that is opened and then clamped on with the help of this loading helicase. It's a very interesting process. Next is, as you know, helicase needs energy. So what molecule does it use? Easy one. What does helicase use to melt DNA and move forward?
Option B.
That's correct. Very good. Very simple question. That it needs ATP. And so it's an energy consuming process where helicase unwinds DNA and moves forward and moves along with the replication fork. All right, so what do these single-strand binding proteins actually do? So I discussed this earlier. Yeah, someone in the comment has got the right answer. Does anyone want to say their answer out loud? Then that sounds about right. It's option A. Very good. So it prevents the single-stranded DNA region from reannealing to itself. So these are little oligomers of proteins that bind there and make sure that that doesn't happen. Great. Okay. All right. So what structural domain is a characteristic of these single-stranded DNA binding proteins? Does anyone remember?
Option B.
Yeah. Anyone have any other answers, or is everyone happy with that? Then that's right. Yeah. It's OB domain. So OB stands for oligonucleotide binding domain. And essentially, what the SSBPs do is they use this domain for 2 reasons. One is that it helps form this kind of groove that allows it to surround the DNA because it interacts in such a way that it allows it to surround it like a loop. And the other is that it helps with oligomerization. So I mentioned that it's just a bunch of these SSBs that are kind of linked to each other. So that's with the help of OB domain as well. So that's the function of that. Moving on to the next question. What is the function of the sliding clamp? I talked about this earlier. You guys remember the sliding clamp? Anyone in the comments? Yeah, someone in the comments has got it absolutely right. Anyone else has any other ideas or any confusions about this? No? Okay, then that's absolutely right. It's A, which is it increases the efficiency and processivity of DNA polymerase. Right, so it's what helps the DNA polymerase attach and stay on the DNA, move along it, and add many nucleotides without making a mistake or without falling off. So great. Okay, question number 31. DNA polymerase 3 synthesizes DNA by adding nucleotides to which end of the primer? So this we covered earlier, but I thought I'll just come back and make sure everyone knows. This is very important.
Option B.
That is correct. So because it's adding to the 3' end, it's like it's growing from the 5' and 3 to 3' direction. So good. I'm glad you guys know that. Right. I think this is like the second last question here. So how many core polymerases are present in the DNA pol3 holoenzyme? So do you remember that image that the professor had shown, where you have one attaching to the leading strand, another at the lagging, one in the middle that helps the cycling? So how many are there? Then there's 3, right? So that's the answer. It's option C, 3. So the DNA polymerase 3 holoenzyme has 3 core polymerases. It has a clamp loader and it has these tau subunits. I think there are 3 of them, 3 tau subunits that have the flexible linkers. I'd shown the image earlier, right? So you saw those linkers in blue. Maybe I can go back. Yeah, here. So you have the 3 core subunits, your tau proteins with their flexible linkers that allow them to help the binding of the clamp to the core. This is the clamp loader, and this is the actual clamp that allows it to loop around the DNA and allows it to move. This, I think this clamp loader has 5 subunits, yeah. So this is how it looks. So it's good to know this because it's important to understand how this entire protein complex, what components it has that allows it to move forward in the way it does. Oh, okay. This is the last question before another interesting thing I thought we could watch together, which is about a different mechanism. So primase activity is regulated by which of the following, which is question number 33. What do you guys think?
Option A.
That's correct. Yeah, that's correct. Primase activity is in fact regulated by binding to helicase. Great. Good. So now there's one more thing that is important when you think about prokaryotes. So we talked about how their chromosome, how their genomic DNA is replicated. It's all in, of course, in the cytoplasm only. It's not like they have a nucleus and something is episomally expressing or whatever. But you have another type of DNA that's present in a lot of these prokaryotes or bacteria, and that is the plasmid. You guys have definitely heard of plasmids, right? And they have various different functions and help out the bacteria in various different ways. They can provide antibiotic resistance. They can provide some sort of way to communicate with other bacteria. So all sorts of different things, and you have them existing homeostatically in bacteria. So how are they replicated, right? Is it the same as the circular sort of covalently closed circular DNA of the genome, or is there something slightly different? So there's this rolling circle mechanism of plasmid replication that was discovered, and there's this short video on that I thought I would show you guys that really nicely sort of visualizes how it happens. So let's take a look at it together. Let me know if you can't hear it.
In this image, the DNA from a bacterium has been gently released from the cell. Most of the DNA is from the bacterial chromosome, but small circular DNA molecules called plasmids also exist. For many plasmids, replication is not tied to chromosomal replication. Many plasmids replicate autonomously by a method called rolling circle replication. In rolling circle replication, a replication initiator protein called Rep-A binds to a section of the double-stranded DNA called the origin of replication, or ORI. Rep-A is encoded by a plasmid gene. Rep-A nicks one strand of the DNA and holds on to the 5' end of the strand. The 3' end, with its free hydroxyl group, serves as a primer for a host DNA polymerase to begin to replicate the intact complementary strand. The Rep-A initiator protein recruits a helicase that unwinds the DNA. As the DNA unwinds, it becomes coated by single-strand DNA-binding proteins. As replication proceeds, the nicked strand, which continues to be covered with single-strand DNA-binding proteins, progressively peels off until replication of the intact strand is complete. The two ends of the nicked single strand are rejoined by the Rep-A protein and released. DNA ligase seals the nick in the double-stranded molecule. The single-stranded DNA can now be replicated. A region of the DNA becomes looped, allowing RNA polymerase access to the DNA to form a primer. Host DNA polymerases use the primer as a starting point for the synthesis of DNA. Finally, DNA ligase seals the remaining nick, resulting in a double-stranded plasmid. Each of these plasmids can undergo replication again by the same method.
Right. Great. So that is about rolling circle replication. I just thought I won't go into too much detail about it, but I thought it would be useful for you guys to see that there are actually two modes in which it uses the same components. You saw that there's DNA polymerase, ligase, but how it replicates, and in fact the timings. It's not always that the DNA is replicating of the main genomic material is replicating, so all the plasmids are replicating at the same time. These, as they said, can be asynchronous, so not happening at the same time. So it sort of depends on what the cell or what that bacterial cell requires at the time. Yeah. Cool. So this was about the prokaryotic replication, and I have some questions and all relating to the eukaryotic one. I think we can take a short break till 7:30, so 7 minutes from now, and then we can resume. Is that okay with everybody? Okay. I'm assuming it's okay with everyone. So this you can remain in the meeting, maybe just chill out for a bit, and we'll be back um, at 7:30 then. Okay. So I think I'll continue with some questions relating to eukaryotic um, replication. So just a few questions. I thought I won't take it too long, and it was explained in quite a lot of detail in the third video of replication, DNA replication by your professor. So I thought I'd just dive right into some questions relating to it. As for any, if you have any questions, please let me know as usual, and you can always post questions on the group, on the common group. So as you know, there are some differences between the DNA replication okay, DNA replication machinery between eukaryotes and prokaryotes. So the eukaryote that has been mentioned in quite some detail is yeast in your coursework, and E. coli or any other bacteria has been taken as an example for a prokaryote. So some examples are, of course, that while the general replication machinery is conserved and the mechanism also is quite conserved, there are the different components are, in fact, I'd say just slightly more complicated when it comes to the eukaryotic version of DNA replication. So, for instance, helicase, while in E. coli it was just DNA-B, in yeast, for instance, it's known as a CMG complex. So you have these different components that are part of one complex, including CDC45, I think MCM27, and one more GINS. So these are there as part of the CMG complex, and they function cohesively with one another. Then another thing you have is you have different DNA polymerases that form the core, not just uh, not just the DNA pol 3 core with its three um, um, replicates that allow the leading strand and lagging strand to be extended, but you also have DNA pol delta and DNA pol epsilon that help with the lagging and leading strand respectively. You also have obviously DNA pol alpha itself, that is the one that's responsible for actually creating the new chains. So it has three different DNA polymerases that are working together at the replication fork. So that's, proving my point that it's so complicated, but it's not impossible to understand. Another interesting point is that the eukaryotic clamp loader. So you know that in the prokaryotic, we talked about the clamp loader, about how it's how the polymerase is loaded onto the DNA. So here, this clamp loader is not bound to the DNA polymerase. And interestingly, even though our DNA replicates quite quick, it is actually so much slower in eukaryotes like yeast, mammals, whatever. It's like about 60 base pairs per second in comparison to E. coli, which, as you would remember from previous slides, was up to 1,000. And yeah, the Okazaki fragment length differs. In prokaryotes, it's a lot longer, it's a lot shorter in eukaryotes, and we will discuss why that's the case with one of the questions. And of course, you have to wonder, why is it that this process is different in eukaryotes versus in prokaryotes? Why can't we just use the same machinery? Right? So there are some reasons. So one is that the eukaryotic chromosomes are linear. So to accommodate linear and much longer, there's so much more DNA. So to accommodate that, we have different multiples of different origins of replication that allow simultaneous replication of different regions that are then sort of put together. And we also, to make sure that the ends of the chromosomes that come from the linearity, so that we don't run into this end problem, we have telomerase enzymes and telomeric repeats at the end of the chromosomes that allow us to replicate all the way to the end, so we don't lose it. And you guys will probably read about this, later on, or, in your free time if you're interested, about how, telomerase and the presence of the enzyme or the lack of the enzyme that can determine aging. Like, not, it's not the sole factor, but it has been sort of, a lot of research has been tied into reading about how aging and senescence is related to telomerase and telomere presence, and or the shortening of telomeres. So how active your telomerase enzyme is can tie in with cell senescence, and also messing around with the telomerase activity in cells can actually um, immortalize your cells. So it can constitutively, if you have a telomerase activity, essentially you will not have shortening of telomeres in linear chromosomes, and that can allow your cells to keep dividing and keep dividing for many, 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 many cycles. So that's how we have these immortalized cells that we work with. These different cell lines that you passage continuously for many, many passages, how are they not becoming senescent? Because many of them have been immortalized. You can use these viral constructs. We have one in our lab called HTERT that allows you to do this. So I thought that's something interesting that you guys can think about. It's not the cure all. You can't become immortal or a superhuman, but it is it's something that's possible, at least on the cellular and tissue level, maybe. Yeah. So now, with that introduction, I'll just move to a couple of questions and then we'll wrap it up for today. Okay. So, which of the following, this is all with respect to mostly, I think, eukaryotic DNA replication. So, which of the following is the the helicase complex in eukaryotic DNA replication? So, I mentioned it just now while introducing. Do you guys remember what I said?
Option B.
That is correct. Very good. So, I mentioned that helicase is, or DNA B of prokaryotes is a little more complicated in eukaryotes because it's a complex of three proteins in the CMG complex, CDC45, MCM27, and GINS. So that's the CMG complex, and it is also similarly regulated um, here as well, where you don't want over activation of helicase. You also don't want helicase activation in the wrong like, you don't want it to bind too many times to the DNA and replicate it too many times or cause single stranded regions too many times. So there's actually a very interesting way in which that binding is regulated, right? Where based on, in your G1 phase or your S phase, based on the CDK levels, you can have a difference in helicase loading and helicase activation. So in the G1 phase, right, which is leading up to the S phase, so your growth 1 phase, the CDK levels are typically kind of low, and that allows helicase loading and then activation in the beginning of S phase, and then later on, SG2 and all, you have high CDK levels, and that inhibits, and then the rest of the um, cycle, essentially you have higher CDK levels, so that prevents helicase from binding again, because you only want to replicate the DNA once in a cycle, right? So that's an interesting sort of check mechanism that that the cells have, to make sure that you're not replicating DNA too many times in the cell in one cycle. Yeah. So what is the function of DNA polymerase alpha? So this also I mentioned earlier while introducing. What do you guys think?
Option C.
Option C. Okay. Uh, Not entirely. So that's not the right option. Does anyone know? So it's actually, I think I have not also written it very nicely, but the function of, one sec. Oh, my bad. I'm so sorry. I was looking at the wrong slide myself. Okay. on my end. It is correct. You're right. It's primer synthesis. You're absolutely right. I was looking at something else. So whoever said that, I'm sorry for confusing you. You're right. It's primer synthesis. I thought I was looking at delta. Yeah. Yeah, great. So moving on from that. So just to clarify my error, the function of DNA polymerase alpha, which is essentially primase of eukaryotes, is primer synthesis. Yeah. Yeah, so in eukaryotic cells, next question, replication occurs during which phase of the cell cycle? This is very easy. That's correct. Person in comments, you're absolutely right. It's S phase, synthesis phase. That I just, it's a given. Okay, what is the sliding clamp protein equivalent in eukaryotes? Okay, Pratik and Samiksha say B. Does anyone have any conflicting answers? Does anyone think it's ORC? No, right? That's great because you're right. It is PCNA, and that's in fact interestingly used as a marker because um, it's a marker for highly proliferating cells. So it's actually been used as a marker while trying to figure out which cells are highly proliferating, say from a whole bunch of cells. You can use it as a marker um, to visualize that. So PCNA is indeed the sliding clamp protein equivalent in eukaryotes. Okay, so this is just a couple of factual questions really, but it's useful to know this to understand the scale of these things. So the replication fork progression rate in eukaryotes, how much is it approximately? What do you guys think? So yeah, this is a bit factual, but anyway, it's good to know. It's around, it's option B, which is 20 to 60 base pairs per second. So think of how much slower it is in comparison to prokaryotes, which go up to 1,000, right? And still somehow we manage to duplicate our DNA. This is crazy. Okay, what is the role of ORC or ORC in DNA replication?
Option C.
That's great. That's correct. It is important for um, helping to bind to the origins. I think others have also got it right. So this is essentially your initiating complex. So it recognizes and binds these different origins. And in the yeast, you have these ARSs, these autonomously replicating sequences, and they are very AT rich, and that's where the ORC. So AT rich, just like how it's in the OD in prokaryotes, that's where the ORC goes and binds. And you have multiple of these like ORI sequence equivalents in eukaryotic DNA. So you have multiple orcs that go and bind. And of course, an interesting point to note that was mentioned in your coursework was that it doesn't have to be that all of these ORIs or all of these ARSs are sort of activated at the same time or firing at the same time. You can have multiple ORIs far apart, close to each other, and some of them might like fire at some phase during sometime during S phase, some later. So it doesn't have to be that all the ERSs that are sort of sequentially there in one region, one after another, are all firing at the same time. It kind of depends, which is very interesting. And a lot of research, I think, is being done about why that is the case and what controls that. How do you control which ORI is firing when? And why is it needed? Why does that have to happen? So can it be because of external cues? Can it be because of internal um, sort of cues that it's getting from some requirement of the cell? So that's something that's being studied. Okay, few more questions I think are left. How many essential DNA polymerases are involved in eukaryotic replication? So I mentioned couple of them. So someone is saying 4, so option D. So I can understand why you're saying option D, because then like you have alpha, delta, epsilon, and then you have alpha. Um, sorry, you have alpha, delta, epsilon, and then you have the entire complex. But actually, there's just 3 of them, right? That are involved in replication. So the answer is actually C. Okay. So in yeast, I was just talking about them, ARSs, they are identified by their ability to.
Option B.
That's very correct. So just from the name itself, you can tell that they can actually initiate replication independently. So as I was saying, how ORC can bind to these ARSs, and it can initiate, and it can fire at different times, and it can help initiate this replication. Yeah, so which enzyme is responsible for loading the pCNA sliding clamp?
Option B.
You'd be right. Very good. So you're completely right. It is RFC. Okay. The ARS elements in yeast contain which conserved sequence? Anyone? So one way to tell is when you have something called a consensus sequence, that will tell you a lot about this answer. So suppose you have several different sequences of say some origin of replication between different organisms, and you want to see whether it's conserved. So how do you do this, right? So you can use this bioinformatic technique called multiple sequence alignment. You can use this NCBI database, the BLAST function, right? So you can align multiple sequences and look at where they are the same or where they are aligning without too much sort of differences or heterogeneity. And after aligning optimally, so your algorithm does that, where essentially it's moving one by one by one by one and checking, okay, these different sequences, how are they aligning? Do they have a consensus sequence, aka do they have regions that are the same across them? So a consensus sequence is the essentially an average sequence that you get from a multiple alignment algorithm to look at what is the how well is it conserved across those different examples. So say you took a human, one gene from a human, the same gene or an equivalent gene from yeast, and another one from bacteria, and another one from say a, a protist or something. Then and then you aligned them, and then you looked at the consensus sequence, which is essentially what is the average sequence that is the same across them. So that is conserved sequence. So in this case, from the name, you can tell itself ARS consensus sequence or ACS. That is the correct answer here. So that is the conserved sequence in the ARS elements. That means it's not changing, and it has not changed in a very long time. Evolutionarily speaking, a conserved sequence is something that has not mutated a lot, has not recombined a lot to become very different over many generations, years, and mutations. It still remains very similar. So I thought that might be useful for you guys. Right. Question 11. So the MCM helicase complex, it's recruited to the origin during which phase?
Option B.
That's correct. Very good. So you're right. It is during G1 phase. And makes sense also because initially you're loading during G1 phase, and then after that, after just one round, you're going to have inactivation. You're going to prevent helicase from binding too much. Someone messaging me? Okay. Cool. Next, which of the following prevents re-replication of DNA within the same cycle? So this we discussed earlier.
Option B.
That's correct. I've somehow put all of the correct ones as B. Anyhow, that's correct. It's high CDK levels in later S and G2 and M phases that make sure that you're not sort of melting the DNA too many times, just the once. So great. I'm glad everyone's understood that. I'm hoping everyone else, apart from this one resilient person who's answering all the questions, I hope everyone else is also understood. And please let me know if you're confused anywhere. Last couple of questions and we're done. So what role does DNA Pol delta play in eukaryotic replication? Anyone? So there is DNA Pol epsilon and DNA Pol delta, right? So they're both, very important for proofreading and they're very highly processive enzymes. So one of them is more for leading strand and one of them is more for lagging. So DNA Pol delta is more for the synthesizing of the lagging strand. So option C. So that's just something good to know because you have these multiple components coming together to replicate the DNA. So each of them has a function and each of them has a way of not hindering the others. So it's option C. Okay. And then what happens when the replication fork encounters supercoiling ahead of it? So someone had confused this one with helicase earlier. So what do you think the answer is, guys?
Option C.
That's correct. Great. So topoisomerases, they relieve what is called, yeah, everyone in the comments too. Very good. It's option C. Topoisomerases, they relieve the what is known as torsional stress. So what would happen if there's supercoiling and a replication fork is approaching it? That means that you have an open region and then you have a region that's very coiled up. So that leads to an actual building up of stress. And you can't have that, right? Because how can you then either you stop or you go on? But if you need to continue, yeah, the person texting now as well, great. Um, if you want to keep moving on, you will have to make sure that some amount of that torsional stress um, in the whatever, like the conformation of the DNA strands ahead is relieved, such that we can continue to melt more DNA and form without it getting really. It's like think of it like a string, I guess. And if you have it like tightly knotted up on one side and you keep pulling the two apart, like from the two ends apart, you're going to end up with a lot of strain. But if you slowly relieve it from one end without actually separating the two strands, that would help with reducing the strain and also allowing the replication fork to move ahead. Right. Which of the following statements about telomerase, which we discussed earlier, is true? So I discussed telomerase earlier. Do you guys remember what I said about how it's tied in with senescence? That might give you a hint. That's correct. Yeah. Someone is saying it prevents shortening of linear chromosomes. So good job. That's the correct one. It helps to synthesize these telomere repeats and make sure that that region at the end of a linear chromosome that's difficult to prime, right? Because you need a primer to add on, but so much close to the end, how do you do it? So that's where these telomeric repeats come in, and telomerase enzyme is useful. And this is the last question for today. What feature of eukaryotic replication helps to complete large genome replication quicker? So someone is saying B. I don't know if it's for this question or the previous one. Anyone has any other answer? Someone is saying A. Disha is saying A. Great. So the answer is correct. That's A. That you mentioned, that you have multiple origins of replication. I mentioned this earlier, so I'm not going into it. So that is it. I think those are all of the questions. I had more questions today than sort of text or stuff to teach you. So apologies for that. Next time I will prepare a few more slides on more interesting and maybe out of the box things um, with more pretty fluorescence images, hopefully, because that is my favorite thing. So I hope you guys understood sort of some of the concepts relating to prokaryotic and some little bit of eukaryotic replication that I covered here. And I hope the questions were useful in clearing up any confusions that you might have had. Please let me know if you have any other questions. Please feel free to post on the group. And yeah, that's it for today. Does anyone have anything they want to ask or say or anything? Any feedback you want to give with respect to what you want to see in the next TA session? If you want me to cover something specifically that was confusing to you before, you can totally let me know. I'll prepare a slide for it. No? Everyone is good? Okay, then I think that's it for today. Right, thank you all for joining, and I'll see you next Monday from 6 to 8.